But we are focusing on late-stage disease, but perhaps the reason I'm in the early session is because it's preclinical at this point. I'll tell you about how we really are pushing microRNAs into the clinic and some of the data that we uncovered for a particular microRNA, in particular miR34A, and where we're at with moving that forward. So just to set up the context, microRNAs were identified 26 years ago, but the first human-encoded microRNA was only identified in 2000. So we're still quite early in the field. And I'm going to come back to this microRNA LET7 later on in my talk. One of the seminal things that happened in the microRNA field shortly after their initial identification was work done by Carlo Croce's group, where he determined that there was a cluster, 2 microRNAs, that their deletion of basically 44 nucleotides in the human genome could be responsible for chronic lymphocytic leukemia. So this argued that not only are microRNAs found in the human genome, but their involvement in disease is also quite common as we began to understand in 2002, and this has been really enhanced over the last decade or so. So a seminal period in the field that's relevant to my talk is in 2007, a family of microRNAs was identified that are downstream of the p53 tumor suppressor, and there's 3 of them in that family, miR34A, B, and C. And I'll come back to that point shortly. But what's really important to know here is that this microRNA family was identified in 2007. We began to understand the biology of what this microRNA family does, advanced it into preclinical animal models for treatment of disease. And in 2013, this microRNA went into clinical trial. So in as short as 6 years from identification to clinical trial. So what I'm going to focus on today is really the work that happened in between here and how miR34 entered into clinical trial. The clinical trial right now is for hepatocellular carcinoma, so either primary or any other type of cancer that has metastasized to the liver. We recently have been awarded a SPOR through Yale and Harvard that's led by Frank Slack and Roy Herbst to actually add lung on as an additional target for miR34. So the data I show you today is going to be what fueled that SPOR. Okay, so microRNAs, as far as their general biogenesis goes, I'm not going to go into a lot of detail, but there is a primary transcript that's expressed in the nucleus. Through the microprocessor complex, it gets processed, exported out to the cytoplasm, further processing by dicer to a microRNA duplex, and the mature strand gets loaded into the RNA-induced silencing complex to ultimately inhibit protein-coding gene expression. Much the same way that proteins can be referred to as oncogenes and tumor suppressors, such is the case for microRNAs. We have microRNAs that, when they're overexpressed, are thought of as oncogenes. This essentially means that their normal role in the cell is to target tumor suppressor genes, and if you have too much of them, you get too much downregulation of protein-coding tumor suppressive genes that could lead to aberrant cellular proliferation. Similarly, you can have tumor suppressive microRNAs where their biogenesis or their expression is lost, and their normal role is to downregulate oncogenic proteins. So if you lose these tumor suppressive microRNAs, you get inability to repress these critical genes that are drivers of cancer. And this is the case for the microRNA that I'm going to focus on today, which is miR34. It's considered a tumor suppressive microRNA. It was identified, as I mentioned, as being downstream of the most commonly mutated gene in cancers, p53. In p53 null tumors, if you simply overexpress miR34, you can induce cell cycle arrest and apoptosis, suggesting that some of the activity of p53 can be attributed to this family of microRNAs. And this is done via a variety of targets that miR34 hits. This is not an exhaustive list, but many of these are involved in proliferation, cell cycle advancement, and so on. So when we think about microRNAs and we talk about targeted therapeutics for any disease, in the context of this group this morning, non-small cell lung cancer, one of the issues, and I think we'll hear about this, is resistance. And we like to consider microRNAs not as a single drug cocktail, but as a multi-drug cocktail. And that's due to the fact that they have multiple targets. That's one of the reasons that we believe microRNAs should be looked at and advanced therapeutically. The chance of resistance to all these targets is relatively low, as we would imagine. So I'm going to tell you some of the data, as I mentioned, that is pushing this forward. So this is the mouse model we're using. It has an activated KRAS allele and a p53 mutation that's analogous to the human R175 mutation. We induce transgene activation in this mouse model via intratracheal intubation, which I'm showing here. So it's essentially a needle that we use for a human blood draw. We intubate the animal much the same way as most of you are clinicians in the audience. This is very similar to human intubation, although it's much smaller. We have a fiber optic light that shines on the thoracic cavity to illuminate the trachea. Mouse is fully anesthetized. And the trachea is about illuminated about the size of a pinhead, if you will. And so that's our challenge, to get that catheter in there. Once we do so, we deliver a viral, in this case, either adenovirus or lentivirus expressing Cre recombinase that will excise these stop cassettes and induce activation of these two transgenes. And so at this point, the viral load is being administered to the animal, and the animal will take that up specifically into the lungs so that we're only inducing transgene activation in the lungs of these mice. So following transgene activation, we look at tumor formation in these animals. And as soon as this video is done, we'll progress over. This is what we see as far as histological analysis of the tumors. We get some mild hyperplasia starting around 8 weeks. That advances in this particular mouse model into adenocarcinoma that is invasive. We get metastatic disease to the liver, kidneys, and lymph nodes. Beyond 19 weeks, we get massive inflammatory response in these animals, which could be a very great model to understand some of the immunotherapy that we're considering in non-small cell lung cancer. But for the purposes of today, anything I show you is going to be terminated at this 19-week time point. The first thing we want to determine if the miR34 levels in this mouse model were low, so we excised the tumors, and all 3 miR34 family members in the tumors of these animals were low. This wasn't the case for all microRNAs. A couple of the oncogenic ones, including 21 and 155, were higher. miR34 target genes at the transcriptional level, we found 2 of them that were upregulated, as we'd expect if we lost that negative regulation. 2 other miR34 targets weren't changed transcriptionally, and unfortunately, due to small amount of tissue available, we were not able to determine if the protein for these was elevated, but likely it would be the case. So once we had some data that argued that miR34 was down and the targets was up, and other data that I don't have time to show you today in cell culture, we took this to the mouse model, and we asked this, our first question was whether or not miR34 could prevent tumor initiation. So in this model, at the same time as activating the transgenes in this mouse model, we delivered into the lungs a lentiviral encoded miR34A, and the animals were sacrificed at 19 weeks. We were very surprised by the data that we received after this study. We were hard-pressed to actually find any tumors in the lungs of the animals that were treated with miR34A. This mouse model has been inherently difficult to treat, so again, highlighting the strengths of miR34 if we deliver it directly to the lung. And this is quantified at the bottom. The section I'm showing you here that has just a small amount of hyperplasia and 1 nodule here is actually the worst tumor that we saw in our cohort. But we all know that people don't typically come into the office looking for an inhibitor of non-small cell lung cancer. They're looking for a treatment. So we challenged ourselves and said, okay, let's first treat the animals, let the tumors progress for 10 weeks, subdivided these into 3 treatment arms. The first treatment group, or the first group of animals, was sacrificed, and that gave us a baseline level of what the tumor burden was at 10 weeks post-transgene activation. The other 2 arms were divided into those getting miR34A or control, and they went an additional 4 weeks. And the data for this histologically look at as such. So the baseline animals, some tumors forming. Again, these are sacrificed at 10 weeks. Both of these treatments, the control or miR34, went an additional 4 weeks. The controls progressed from 10 to 14 weeks as we'd expect. The miR34, basically no progression at all in those. We weren't able to determine if we'd have regression because the initial baseline tumor burden was low. We're trying to repeat some of these studies using CT scanning or MRI for animals so that we can actually follow tumor burden in individual animals instead of having this baseline sacrifice group. Regardless, this suggested that perhaps miR34 was going to be a valid therapeutic option, but we decided to challenge ourselves even a little bit further. And I mentioned this pathway already with miR34 downstream of P53. What I didn't mention to you with regard to microRNAs was the KRAS pathway, and this is just one arm of that, but as all you know, this is a highly activated pathway in non-small cell lung cancer. The first human-encoded microRNA that was identified by Frank Slack and his group was LET7, and that targets RAS. So our argument was perhaps we can combine miR34 with LET7 to target P53 and KRAS-activated cancers. And like miR34, LET7 has other targets independent of RAS, including other genes involved in oncogenesis, HMGA2, LIN28, and MYC. So in addition to challenging ourselves with this combinatorial therapeutic approach, we also even moved to a more aggressive mouse model that I'm showing you here. So this is the model that we had last time, KRAS with the R172 allele. And on this side here is KRAS with the P53 floxed allele, so completely knocking out P53 in the lungs. These are both showing 8 weeks, so you can see that this is a more aggressive mouse model. This up here is just showing you the targets of LET7 and miR34 in the context of some of the most activated oncogenes in non-small cell, and of course P53 somewhat restored by miR34. So we believe that this combination would be hitting multiple relevant targets in this disease. And then finally, we partnered with Mirna Therapeutics to deliver these systemically, so something that's more clinically relevant than lentiviral delivery right into the lungs. So the agent that we are using is MRX34. This is the one that is now in the clinic. It's basically a microRNA mimic encapsulated in an amphoteric liposome that basically changes its charge in the acidic environment of the tumor so that it's more readily uptaken by the tumor cells relative to non-tumorigenic cells. And so this mouse, this is essentially how our preclinical analysis was. We activated transgenes for 7 weeks and had the various treatment arms of control, LET7 alone, miR34A alone, and half the dose of each. And dosing was at 1 milligram per kilogram. The reported toxicity for this is 27 milligrams per kilogram, so we're well under the toxic dose here. And this was every other day for 28 days. So as far as safety profile in this mouse model, we didn't see any elevation of cytokines other than in our LPS control. So none of the other ones, IL-1 beta, IL-6, or TNF-alpha were elevated. We did see accumulation of the microRNAs in the lungs, shown on the left-hand side here, and on the right-hand side in the whole blood, just showing you miR34 in this case. In both situations, when we treated with half the dose, we got approximately half the amount of RNA present. But what did we see therapeutically? These are the animals that were treated with the negative control, again, much more aggressive than the last mouse model. LET7 alone, miR34A alone, or half the dose of both. We saw statistical reduction in the tumor burden, which we quantify as tumor relative to total lung area, and the size of the tumors was statistically smaller in the combination therapy here. We took this to a survival study, and unfortunately, and somewhat fortunately, but unfortunately what we saw was that the dual-treated animals lived at the same, had the same survival benefit as animals administered miR34A. The difference with this is that we upped our dose to 10 milligrams per kilogram, and likely what was happening in this study was that miR34A effect was swamping out the LET7 effect. We actually saw no therapeutic effect at all with LET7 alone in this mouse model. So we see a 40% increase when we treat with miR34A, which is as good as any other FDA-approved agent in this mouse model. And this was a single trial, dosing on the bottom here, and a single dose. So that argued that, again, perhaps miR34A is the microRNA that we should move forward with. And this leads me to some of the current work that we're doing in the lab, which is how do we enhance miR34A to get ready to basically take this new miR34A enhanced therapy into the clinic, hopefully once the miR34A monotherapy gets approved. So we combined miR34A with every single human-encoded microRNA in the human genome at that point in time, which was 2,000. These were tested with half the dose of miR34A and half the dose of the other microRNA. Each of these dots represents one of those tests and basically relative proliferation of non-small cell lung cancer cell line. Black line represents the control. The full dose of miR34A, i.e. miR34A plus miR34A shown here. And the red lines represent 3 standard deviations on either side of that. And we came up with 2 groups. We came up with a group of microRNAs that perhaps may antagonize or prevent miR34 activity, and a small group on this end, but still 300 microRNAs that might act synergistically or additively with miR34A. When we performed this, we did the microRNA in combination with miR34A. We never tested the microRNA on its own. So of course we needed to ask, are these acting cooperatively with miR34A or independently? And so That's shown here. This is basically a mathematical model of what we would expect. If the microRNA complex with miR34A was better than the microRNA complex with itself, we'd expect a score of less than 0 in this model. Anything else would argue that the microRNA was not acting cooperatively with miR34A. So we tested these 333 microRNAs. We found some that were likely monotherapeutics, which I won't discuss. There would be in this group. Those that were less than 0 are shown down here, and that was 112. So overall, not to get into the details of this, what we determined was that out of these 2,000 microRNAs, we narrowed it down to 331 that seemed to act with miR34A. Of those, 115 were cooperating. We've since tested this in a variety of other cell lines and have narrowed it down to 55 microRNAs that are active in either 5, 4, 3, or 2 out of 6 cell lines. And we're proceeding forward with 10 of those. And of those 10, we're identifying the targets for them using a somewhat novel approach where we're pulling down the microRNA bound to its target and doing a simple ligation of the two so that we can actually find out what target is that microRNA binding to and understand the biology of how they're synergizing, as well as perhaps what are we hitting that may be a valid therapeutic target with these microRNAs. And so I won't go through the conclusions. I do, however, want to acknowledge my lab. I am not doing a lot of this work. That's really the work of a very talented graduate student, Esteban, in my laboratory, and an undergraduate that actually works with him. Of course, some of this work was fueled by Frank Slack's group when I was a postdoc there, and our collaborators at MicroRNA Therapeutic, who are taking MRX34 forward, and some of the other work that I didn't have a chance to talk about, and some of our other collaborators. I'd be happy to take any questions that you have.
Not sure if this is a question or comment, but miR34, I actually think of it as being a cell differentiation microRNA with high levels in early differentiation of airway epithelium. So I'm not sure if you've thought about that. It's more specific to early development as opposed to later differentiated in sort of it's present in basal cells in airway epithelium. So that's a comment. The second is that miR34 has the same seed sequence as miR499 B and C. So I was wondering if you looked at those as well.
No, so both of those are good points. No, I have not looked at miR34 levels in the early epithelium. Predominantly, everything we're looking at is in later stage cancer, and it's a valid point, though, what it's doing there. I'm not familiar with. As far as the other miR34 family members, yeah, there's a triple knockout of the 3 miR34 members, A, B, and C. It has no phenotype. It's been reported by Andrea Ventura, likely because there are other families that we weren't aware of that have since been identified that are playing a similar role. I think the experimental approach that we're doing right now, where we're looking for the targets of these, doing this clash protocol and pulling these down, is going to identify other family members that may be not binding based on the canonical seed sequence, and maybe our understanding of microRNA families isn't as developed as it needs to be. So these are very valid points in the field. We need to understand that before too much clinical advancement.
So really quick question. MicroRNAs have also been implicated as immune suppressors of activated T cells, and particularly LET7. So you're probably aware of the work of Teresa Whitesides, who's shown that microRNAs released by exosomes from tumosomes and circulating in the blood can be easily detected. So the question is, do you think that we could use that as a biomarker by looking at serum exosome content of microRNAs to try to identify what type of activity is actually needed for patients?
Yeah, so I'll kind of divide that into two sections. So the first one is, I think for about 4 or 5 years now, there's been some data out that argues that a 10 microRNA signature in the serum, I don't know if it's specifically in the exosomes, but in the serum could be a biomarker for non-small cell lung cancer and detect individuals with early stage disease prior to the CT scan that's been done, the CT scan that's been done for patients at risk. And so during that study, serum was extracted from these individuals and blood samples was taken, and a microRNA signature could detect it 6 months sooner. So I do think that these biomarkers, microRNA biomarkers in circulation, are certainly going to be capable of detecting disease. The problem is that they miss people, for one. And then the second part of the question is, will they predict treatment or what's going on? I don't know the answer to that. Some of the cells are probably releasing microRNAs that they don't want, and so that will allow that cell to grow. And then in other contexts, yes, and these exosomes are oncogenic microRNAs that are probably helping the microenvironment as well as opening up the blood. We actually have seen that microRNAs are being released and allow for extravasation into the bloodstream. So yeah, active area research, good questions for sure.